Welcome back to Bio 111. This is chapter 16. We're looking at section 4, eukaryotic transcription and gene regulation. All right. When we look at eukaryotic transcription, it's much more complex, requires a lot of proteins, the involvement of a lot of proteins for it to be initiated, for it to be started. All right. Now, the big actor in transcription is polymerase II. Right? These RNA polymerases require a transcription factor, some sort of specialized protein, to initiate that transcription, to allow it to bind, to allow the polymerase to bind to the DNA. All right? These transcription factors, they bind to the promoter sequence. So it's a sequence of DNA ahead of the gene, and other DNA regulatory sequences. These are sections of DNA that are ahead of the gene that basically are a landing platform for all of these proteins that initiate transcription. All right? Now, one of these regions that the transcription factors bind to is the promoter region. All right? And it can be a few nucleotides, up to hundreds of nucleotides long. Within the promoter region is something called a TATA box, and it's a series of thymine and adenine, thus the TATA name. Within the promoter, just upstream of the transcriptional start site. All right, so what you're looking at, all those little ovals represent some sort of protein that is bound to the DNA to initiate transcription. All right, so this particular transcription factor binds to the TATA box. It recruits additional transcription factors and forms a complex of proteins. The RNA polymerase, the one that's doing the real transcription, it can now bind to the upstream sequence. It can now bind to the DNA that will be turned into proteins. RNA polymerase is then phosphorylated, and part of the protein is released from DNA. All right. It is now in a proper orientation to actually start doing its job and transcribing RNA from this DNA. All right. So an enhancer is a DNA sequence that promotes transcription. Right? Just a sequence of DNA somewhere in the genome. Each enhancer is made up of short DNA sequences called distal control elements. Activators bind to the distal control elements and interact with mediator proteins and transcription factors. Note that two different genes may have the same promoter, but different distal control elements, enabling differential gene expression. And one thing I want you to look at here, distal, we're talking about far away. If you notice, in this case, the DNA has to loop back, bend back in on itself, so the distal control elements can interact with the same factors that are interacting with the gene of interest. All right? Very complex. That's it for section 4. We're going to be looking at section 5, post-transcriptional gene regulation. All right?